Looking at this semi-arid landscape in Uganda, we see a harsh reality for agriculture. When temperatures rise and regions go months without rain, securing a reliable food supply becomes a massive challenge. Under these intense conditions, standard crops struggle. On the left, a bean plant wilts in the dry soil. But on the right, under the exact same glaring sun, a maize plant thrives. The soil and the sunlight are identical. The reason the maize survives while the bean plant dies comes down to the microscopic architecture inside their leaves. This rotating 3D model shows the internal structure of a healthy chloroplast, the engine of plant growth. We can clearly see two primary zones, the coin-like stacks called the thylakoids, and the fluid surrounding them called the stroma. The thylakoids are the site of the light-dependent stage of photosynthesis. This is exactly where the plant uses chlorophyll to capture the sun's energy. Once that light energy is captured, it transfers into the fluid stroma. The stroma handles the light-independent stage. For standard plants like beans, known as C3 plants, this fluid is the primary food factory. This cross-section shows the core vulnerability of those C3 plants. When the weather gets hot, the plant must conserve water, so the microscopic stomata pores on the bottom edge of the leaf pinch closed. Because the pores are shut, oxygen gets trapped inside. Zooming into the stroma, we can see the consequence of that trapped oxygen. The rising oxygen levels confuse the chemical processes inside the chloroplast. The plant actually starts burning energy instead of building food, grinding its growth to a halt. Now look at this cross-section of a C4 leaf, like our successful maize. To solve the oxygen problem, C4 plants evolved a specific structure called Cran's anatomy. Notice the two distinct rings, the outer mesophyll cells and the inner bundle sheath cells. These outer mesophyll cells act as a shield. They exclusively handle the light-dependent stage, capturing the sun's energy while actively blocking oxygen from going deeper into the leaf. Because the oxygen is blocked, the inner bundle sheath cells are protected. Hidden away inside, they can conduct the light-independent stage efficiently, safely isolated from the outer environment. By physically separating the light capture from the food production, C4 plants prevent energy waste completely. They continue to grow rapidly even when the temperature spikes and water is scarce. Let's test what we've covered. Here are two microscopic leaf models. Pause the video, identify which diagram shows the C4 plant, and point out its key structural advantage. If you selected model B, you're correct. The prominent inner ring of bundle sheath cells is the defining feature. Let's look at exactly what is being manufactured inside that protected space. Inside the stroma of those protected cells, a light-independent reaction synthesizes several crucial intermediate molecules. This flowchart breaks them down. The plant produces energy carriers like ATP and NADPH and a highly versatile molecule called G3P. G3P serves as the central branching point for plant nutrition. From this one molecule, the stroma synthesizes simple sugars, vital lipids, and the essential chemical scaffolding for proteins. Understanding this microscopic biochemistry is essential for agriculture. The continuous production of G3P, lipids, and proteins under severe heat is exactly what creates the dense nutritional profiles of drought-resistant staple crops across Uganda. Agricultural officers use this exact biological data when planning for the future. On this map of Uganda's drier agricultural zones, maximizing yield requires recommending specific crops that utilize the C4 pathway, like maize, sorghum, and millet. Looking at this historical harvest, we see that for generations, human populations have relied on the optimized, drought-resistant pathways of C4 crops to feed communities when conditions are harsh. This summary table consolidates what we've learned. Notice the direct comparison. C3 plants thrive in cool, wet environments, while C4 plants divide their labor to dominate hot, dry climates. Inside both, the thylakoid handles light, while the stroma drives the dark stage, turning G3P into essential lipids and proteins. Take a screenshot of this guide to use during your upcoming physical modeling activity, because mastering how a plant builds its food at the microscopic level is the first practical step towards securing a nation's food supply.